So I will start, and it is my great pleasure to introduce my friend and colleague, Dr. Eli Adashi, who is an academic physician and the former dean of medicine and biological sciences at Brown University. He is presently serving as a tenured professor of medical sciences at Brown. He graduated from Sackler School of Medicine of Tel Aviv University and trained in OBGYN at Tufts and in reproductive endocrinology at Johns Hopkins, a program that was led by Professors Howard Jones and Georgiana Jones. Harvard educated in healthcare management with a master's degree. Dr. Adashi is a member of the National Academy of Medicine and the Association of American Physicians, as well as a fellow of the American Association for the Advancement of Science and of the Hastings Center. Dr. Adashi has been the recipient of continuous NIH funding from 1985 to 2005, inclusive of a US Public Health Service Research Career Development Award. NIH service includes the National Council of the National Institute of Child Health and Human Development, the Reproductive Endocrinology Studies Section, and the Selection Committee of the Reproductive Scientist Development Program. He mentored over 50 postdoctoral trainees and is the author or co-author of over 450 peer-reviewed publications and over 120 book chapters and reviews. Dr. Adashi edited or co-edited 16 books in the general area of reproductive medicine, and his work has also been seen press with the New York Times, the Washington Post, the Boston Globe, the Huffington Post, and other media outlets. Dr. Adashi is the recipient of many awards, including honorary degrees from the Poznan University of Medical Sciences in Poland and the University of Ottawa in Canada. Today's Grand Rounds are part of our bioethics Grand Rounds and are hosted by the Montefiore Bioethics Center. And without further ado, it is my great pleasure to invite Dr. Adashi to give us his talk titled Eggs and Sperm from a Buccal Smear.
Thank you, Dr. Tamer, and thank you, all of you who are on this virtual medium, which I certainly am still trying to get used to. I would have rather been there in person and seen all of you in an auditorium. The subject matter for today is, of course, futuristic and entirely disruptive in nature, but as such, I think is very useful. And since it's quite possible that it will transpire before too long, having a discussion ahead of time is always a good idea. I should say that the derivation of gametes from stem cells is really but one component of a very large field wherein stem cells serve as the starting point for a variety of cellular lineages. For example, there are efforts underway to procure amacrine neurons with the retina in mind, potentially dopaminergic neurons with the striatum in mind, trophoblast precursors, motor neurons with paralysis in mind, of course, and gametes, which happens to be, of course, within the domain of my special interest, and in some respect is sort of leading the pack. And from that point of view, is probably useful to review. Many of you probably have heard about the Nuffield Council on Bioethics, which is the premier ethics organization of the United Kingdom, which annually issues an infographic, such as this one, titled, "What's on the Horizon for Bioethics?" And it's interesting to note that under human reproduction, the leading subject matter in this infographic is indeed in vitro derived gametes, which attests to the fact that the Nuffield Council on Bioethics certainly attaches significance to this possible development. Of course, there are many other issues that the Nuffield Council is interested in: time limits on maintaining embryos in culture, womb replacements, preconception screening, whole genome sequencing of embryos and fetuses, genome editing of gametes and embryos, and egg freezing. In vitro gametogenesis is, of course, by its very nature, a disruptive technology. The question is it achievable in the human? And is it really on a short timeline, which means that we might be surprised. And I'll come back to that point, obviously, throughout our discussion today. All of this, of course, harkens back to the classic work of Aldous Huxley and his 1932 book, Brave New World, wherein he predicted that eggs would be produced via ex vivo ovarian cultures, that embryos would be derived via in vitro fertilization, and that newborns will in fact see the light of day via ex vivo ectogenesis. Well, he was, of course, spot on with respect to embryos via in vitro fertilization, which has now been with us since 1978. It's probably less likely that eggs will be finally derived via ex vivo ovarian cultures for a variety of technical reasons. And it's equally uncertain that pregnancy will be exteriorized, if you will, one day, thereby seeing the delivery of newborns through ectogenesis. That said, I should say that there are significant ongoing efforts to simulate the uterine environment, particularly with premature infants in mind, who need to be sustained from 20, 21 weeks onward, and for whom some form of ectogenesis might be appropriate. But going now straight to the issue of today's discussion, in vitro gametogenesis, let me just say that it was briefly alluded to in Brave New World in the following fashion. Specifically, if you recall, those of you who read the book, in chapter 1, the director of the so-called hatcheries and conditioning is conducting a tour of the laboratory, in the course of which he says, 1 egg, 1 embryo, 1 adult, normality, making 96 human beings grow where only 1 grew before, progress. Of course, once we get through our discussion today, you will realize that 96 is really barely an approximation of what will be possible with in vitro gametogenesis. That would be represented simply in a 96 well-played construct, of which an innumerable number could probably be derived. For the longest of time, the possibility, or even thinking about the possibility of in vitro gametogenesis, was precluded by the so-called soma germline barrier hypothesis. That hypothesis was formulated by August Weismann, professor of zoology at the University of Freiburg, who in his 1893 book, The Germplasm, A Theory of Heredity, formulated the so-called soma germline barrier hypothesis, according to which the germline, which we know gives rise to the soma, is by dint of that immortal. On the other hand, the soma, which never gives rise to the germline, or so we thought, is by dint of that mortal. This very influential hypothesis for the longest of time precluded even the notion or the thinking of the possibility of some form of gamete formation from a somatic source. All of that, of course, changed when we were introduced to somatic nuclear reprogramming. The notion whereby you can take a differentiated somatic cell, convert it to a pluripotent stem cell, and then guide that pluripotent stem cell along a gametogenic path to mature gametes. The first technology that made that possible was somatic cell nuclear transfer, or SCNT for short, which, as you know, dates back to 1958, and to the contribution of Professor Garden from Cambridge University, who at the time published a leading paper in Nature titled Sexually Mature Individuals of Xenopus laevis from the Transplantation of Single Somatic Nuclei. What was specifically accomplished was the transplantation, if you will, of a somatic karyoplast, a nucleus, into an enucleated oocyte, so-called oocytic cytoplast, reconstituting a de facto zygote, which was allowed to then develop into a blastocyst, from which Professor Garden derived embryonic stem cells. In 2006, a new technology emerged, courtesy of Shinya Yamanaka from Kyoto University in Japan, who published a leading paper in Cell titled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. In this case, a somatic cell was transfected with select transcription factors, which converted it to a so-called inducible or induced pluripotent stem cell. As you likely know, both Professor Yamanaka and Professor Garden received the 2012 Nobel Prize in Physiology and Medicine for this discovery, which undergirds our conversation today, that is to say, the very feasibility of taking a somatic cell down the gametogenic path. So in vitro gametogenesis, in essence, involves a starting material which would be a somatic cell that would have to be converted to either an embryonic or an induced pluripotent stem cell, which would then be guided towards a gametogenic fate. What would be the prerequisites for making such a transformation, a remarkable transformation, really possible? Well, you would have to know the floor plan. You would have to know the germ cell specification pathway. For one, you'd have to know what are the cues that will be required to convert a somatic cell into a gamete. And you would need to know what is the sequence by which those cues need to be applied and the timing thereof. And finally, you would need to know whether or not there is a so-called niche requirement. That is to say, can this occur in a vacuum, or does it require additional somatic cell types which contribute to the process? And as you, of course, know, oocytes develop in the company of granulosa and theca cells in the ovary, and sperm, on the other hand, develop in the company of Sertoli cells in the testes. And so the likelihood is that a niche does exist with respect to the development of germ cells, and one would have to take that into account. The beginning of this field, best we can date it, goes back to 2003, when Professor Hans Schöller, now at the Max Planck Institute for Molecular Biomedicine, previously still at the University of Pennsylvania, published a science paper titled Derivation of Oocytes from Mouse Embryonic Stem Cells, a depiction of which is shown in the right lower hand corner of this slide. That same year, Toshiaki Noche from Japan published in PNAS a paper titled Embryonic Stem Cells Can Form Germ Cells in Vitro. Only a year later, George Daly from Harvard published in Nature a paper titled Derivation of Embryonic Germ Cells and Male Gametes from Embryonic Stem Cells. And René Rehopera, then at UCSF, published a paper titled Spontaneous Differentiation of Germ Cells from Human Embryonic Stem Cells in Vitro in Human Molecular Genetics. In the first decade of the century, somewhere between 2000 and 2010, several leading publications saw press which defined the germ cell specification pathway in the rodent. Those papers saw press in Nature, Science, and Cell, and were largely contributed by Doctors Azim Surani from Cambridge University, Mitinori Saito from Kyoto University, and others. In the subsequent decade, between 2010 and 2015, let's say, more information began to emerge about the germ cell specification pathway in the human. Again, leading papers in Nature, Cell, and other leading journals appeared that began to clarify what ingredients go into the process in the human context. And so by the time we got around to about 2015, the state of the art was that the germ cell specification pathway is clearly not conserved between the rodent and the human. In other words, merely studying it in the rodent will not do. We would really need to have a solid understanding of how this works in the human. The same held true for the genetic networks involved, which are quite distinct as well. Looking at it schematically, the state of the art in 2015 was that we knew, of course, how to create embryonic stem cells and induce pluripotent stem cells, and we knew enough to convert them to so-called primordial germ cell-like cells. However, we could not take those cells further towards a haploid post-meiotic mature gamete. In a word, we were able to recapitulate the extragonadal component of gametogenesis, but were incapable of recapitulating the intragonadal component thereof. The final frontier, which had yet to be conquered, was, of course, in vitro gametogenesis all the way, not just to the degree that was accomplishable in 2015. Inklings of that possibility became apparent in 2016 when Professor Hayashi from Kyushu University in Japan published a Nature paper titled Reconstitution in Vitro of the Entire Cycle of the Mouse Female Germline. The starting material of Professor Hayashi were, of course, embryonic stem cells or induced pluripotent stem cells that were readily converted into primordial germ cell-like cells. He then proceeded to recreate the niche by mixing these cells with gonadosomatic cells, which led in vitro to the formation of primary oocytes, eventually M2 oocytes, a photographic depiction of which is shown on the left lower hand of this slide, which upon fertilization gave rise to an embryo, a blastocyst, which upon transfer to a surrogate mother gave rise to pups. And so in 2016, at least in the context of the rodent, it seemed that we finally were getting closer to the possibility of generating functional fertilizable gametes in vitro that actually give rise to pups. In 2018, Mitinori Saito from Kyoto University published on the pages of Science a paper titled Generation of Human Oogonia from Induced Pluripotent Stem Cells in Vitro, trying to simulate the same findings that Katsuhiko Hayashi reported in the rodent. However, we are not quite there yet. Professor Saito began with human primordial germ cell-like cell induction, which we know how to do. He proceeded to create or recreate the niche, but in the absence of human cells that would allow him to create this environment, he had to rely on rodent cells, thereby producing what he referred to correctly as a xenogeneic ovary. He then proceeded to allow this mix to develop, and it went as far as producing oogonia. But it never did go beyond that stage and certainly did not complete female determination and or meiotic initiation. The last study I will cite today, just so press in essence, in 2021 in Nature, again by Professor Hayashi, titled Reconstitution of the Oocyte Transcriptional Network with Transcription Factors. In other words, he took the process now to the subcellular level and in fact to the genetic level and identified the transcription factors that are operational in the process. Specifically, he was able to identify 8 transcription factors for converting primordial to primary follicles. And by merely, I say merely in quotations, expression of these transcription factors, converted pluripotent stem cells into oocyte-like cells that fertilized and cleaved. In terms of the terminal human gametogenesis, which we have yet to achieve, the missing links are knowledge of the germ cell specification pathway. We may need to generate gonadosomatic cells or to find another way to simulate their input so as to create a niche. Using rodent cells for this purpose seems less than satisfactory. And one would have to demonstrate in the human faithful replication of meiosis. The latter point has been carefully addressed by several experts from the Jackson lab, who in a Cell paper in 2014 laid down essentially the gold standards for what would qualify as the successful negotiation of meiosis. They require that the resultant germ cells, the in vitro derived germ cells, have the correct nuclear DNA content, have the normal chromosome content and organization, engage in appropriate synapsis and recombination, and give rise to viable euploid offspring. And so if this ever comes to pass in a human, and if the FDA then gets to evaluate the process, they are all but certain going to insist that these 4 criteria are rigorously met. This is where we stand today, if you will, scientifically. And as you can tell, it's close, but not quite there. And so we are left with a circumstance whereby we know this is likely feasible. It might happen rapidly, it might not. And I'll come back to that point because it would be helpful for once to be prepared for a disruptive technology such as this one ahead of time, rather than play catch up. But the scientific and translational promise of this technology is substantial. In terms of the science, this would provide an inexhaustible supply of germ cells. It would allow for functional non-mutant analysis. And it would allow us to study the germ somatic cell interactions, about which, frankly, we know way too little today after years of investigation. From a clinical point of view, this could potentially reverse germ cell failure. It could provide autologous gamete replacements in individuals who, for one reason or another, are unable or are born absent gonadal material. It could be a fine substrate for gene editing, perhaps a superior substrate to the embryo. And in case of genetic disease, this could potentially allow one to correct the defect in vitro. You could even imagine this serving as a source of donor mitochondria for the technology known as mitochondrial replacement therapy, which is designed to correct prenatally the heritable mitochondrial DNA diseases. There will also be a substantial impact of this technology on IVF. This will eliminate the need in the provision of hormonal stimulation of women and egg retrieval. It will do away with the idea of gamete donation. It will convert IVF essentially to a laboratory procedure. It may still require some endometrial preparation and a transfer of an embryo. And in principle, it could improve affordability and thus access. I say so in principle because we always seem to find a way to jack the price up somehow. But when you stop and think about it, the IVF enterprise today in the United States comprises approximately 500 clinics. There is a discipline that's all but dedicated to it, the discipline of reproductive endocrinology. And if in vitro gametogenesis comes about, you have to wonder what in time will happen to this enterprise, which is exactly what happens when disruptions happen. It's very likely that IVF will become a laboratory procedure, which would mean that much of the current infrastructure will not be necessary. And the very discipline of reproductive endocrinology will have to reevaluate its mission going forward. If this ever comes to pass, as I already mentioned, this will have to undergo FDA jurisdiction, most likely by the Center for Biologics Evaluation and Research, which is presently headed by Dr. Peter Marks. It's the same center which approves the COVID-19 vaccines, as you know. And this will likely be treated as what the FDA refers to as HCT/Ps, human cells, tissues, and products, simply because it falls under that category, but it will be rigorously evaluated for its safety and efficacy, for meeting the criteria that I elaborated earlier, and more. This is not going to be a trivial undertaking. What I haven't mentioned, but will doubtlessly will have to be addressed as well, is, are these in vitro mature eggs entirely comparable in terms of their epigenetic makeup? Are they entirely the same in terms of their imprinting makeup? All of those things would have to be assured before we can let them loose and allow human beings, in effect, be created using those substrates. Inevitably, with any technology of this nature, there will be significant ethical considerations to contemplate. On the one hand, there is a Hippocratic beneficence here in the sense that, as I mentioned, there will be clinical benefits. And in some cases, this will have a rather substantial positive effect on the fate of affected individuals. This could certainly play into infertility relief, which again would be a beneficent action that you can envision from this development. But on the other hand, and that's not the first time that argument has been raised, this clearly commodifies reproduction in a way that we have never really envisioned before. The numbers that we're discussing here and the in vitro nature of it all really make it very different than what nature, shall we say, originally intended. Notions of parenthood may well be modified as well, although I will leave this up to your imagination, but a number of possibilities, of course, exist that wouldn't exist when real people are involved. And as is true of any one of these technologies, particularly if they are subjected to genetic engineering, there is what I refer to as the enhancement overhang. Are these gametes going to be used for genetic enhancement and the creation of human beings with properties that may or may not be blessed by all of us at any given time? We are talking also about a magnitude that is previously unthinkable. I'm showing here a 96 well plate, but there would be hundreds of those in principle. There is really no limit on the number of in vitro derived eggs and sperm. And the question would be, can we wisely select, control this bounty? Can we, in fact, act responsibly and ethically to make sure that this does not get out of hand? Limits on scale, limits on choice, and of course, disposition of embryos, which is already an issue even with IVF, are all issues that will come up and will require some form of solution. And I'll say more about that in a minute. This would require some form of serious deliberation by an appropriate body. And potential deliberative bodies in the US that could take that on would include the National Academy of Medicine, perhaps the Presidential Commission for the Study of Bioethical Issues, which would have to be created because there was none in the previous administration, and or the Hastings Center, which is our answer to the Nuffield Council on Bioethics. There are even legal implications here, although time precludes us from a deep dive into that possibility. But just imagine this unthinkable hypothetical outcome, such as unwitting non-consensual parenthood. Imagine that somebody lays their hands on some skin cells that you may have left behind, and for reasons that cannot be justified, will see to it that they are converted into germ cells. And before you know it, fertilization occurs and a human being is born, and whether you knew it or not, you are now an unwitting, non-consensual parent. I know this sounds far-fetched. I know this is probably unlikely, but it gives you, I think, a sense of the kind of, I don't want to say Wild West, but the kind of enormity of possibilities that arise when a technology such as this materializes and is becoming a reality. Because of all of this and more, the public needs to weigh in. This is a relatively new but not novel concept, the idea of participatory public engagement, which has taken hold now, I think, certainly in Europe and increasingly so in the United States. Much of this, as you know, dates back to the 1975 Asilomar Conference on Recombinant DNA, which, important as it was attended by experts by invitation only. The public was not invited. The public was not asked for its opinion. This raised a variety of negative responses, beginning with a speech given by the late Senator Kennedy at the Harvard School of Public Health on May the 9th, 1975, wherein he said, "When science develops techniques that fundamentally change society, society has the right to determine how the technique is to be used, whether it should be developed in the first place, and if so, under what constraints." Senator Javits, not long after that, articulated similar sentiments in a different meeting in Virginia. The Institute of Medicine in 1995 came out with a report titled Society's Choices, led by then Harvey Feinberg, wherein, again, the notion of public engagement was heavily emphasized. And more of the same appeared in 2008 when the National Academies issued a report titled Public Participation, in this case in Environmental Assessment and Decision-Making, the point being that public participation is intrinsic to democratic governance. And finally, Sheila Jasanoff of the Kennedy School of Government and Harvard Law School, in her 2012 book, Science and Public Reason, said that the issue is no longer whether the public should have a say in technical decisions, but how. Clearly then, this all would have to somehow transpire. And if we had our way, we would do all of this before the expected science paper hits the press, where it actually accomplishes the task all the way, and all of a sudden we are faced with a new reality which we haven't really considered. It is in this context that the National Academy of Medicine is considering, as we speak, to begin with a virtual workshop on the subject, to at least begin the process, and later on follow that with a consensus committee report, provided the funding can be secured. These are difficult days in terms of funding because so much of it understandably goes to the COVID-19 effort, but it's possible that in 2022, such a consensus study will transpire. And hopefully that would still be in time before, as I said, the news hit the press. The Brits are in many ways ahead of us on this issue. Here's a nice report that they issued in 2018 on genetic technologies, which involve public dialogue and opinion garnering. And the Nuffield Council, with which we started, in its report on genome editing and human reproduction, called for the establishment of a separate body or commission independent of government, which would produce an understanding of public interest through promotion of public debate. As you can see, the notion of engaging the public is a recurring and now dominant theme, and in my view, a very warranted one. Challenging as it may be, costly as it may be, time-consuming as it may be, but ultimately it's about all of us. And I think in making that investment is probably wise. Which brings us full circle to where we started, and that is the infographic of the Nuffield Council on Bioethics, which placed in vitro derived gametes at the top of its deliberative agenda at the time. Whether or not, of course, this will transpire, nobody can predict with certainty, which is why I conclude with a quote from a poem by W.H. Auden, the Anglo-American poet, who in his poem, If I Could Tell You, says, "Time will say nothing, but I told you so. Time only knows the price we have to pay. If I could tell you, I would let you know." Which brings me to the conclusion of our presentation, and for me to thank you for the opportunity and to see if there's any way I can further clarify some of these issues in the time remaining. Thank you again.
Thank you, Dr. Adashi. Please type in your questions in the chat.
I do not have access to the chat, so maybe you can convey that, if that's okay.
So I wanted to ask you about the public involvement and freedom of science, because let's say that in a certain country, the public decides that mRNA vaccines should not be developed. They're too dangerous. And it is sometimes very, very highly technical technologies. Even the classical recombinant DNA technology, there was huge opposition to it back in the early 70s with a lot of fear, just simple technology, not even PCR, just restriction enzymes and cloning. So how do we balance the need for the public to determine how public money will be used? Because most of this research is paid by the public, and what's good or not good for society with the freedom of science?
That's an excellent question. And of course, no perfect answer, but I think experience has shown that the forums wherein such issues are discussed, and this is known often as deliberative democracy or deliberative democracy exercise. It's not, we're not talking here about a poll. We're not talking here about sampling of opinions by phone, for example. We're talking about the gathering of representative groups of people with experts, which means that the information that the public needs to know is conveyed, hopefully in terms that they can relate to. Not only that, time is allowed for opinions to change, for give and take. This is not a short session, which means that it's also an expensive undertaking. But we're gaining more and more experience with this. And even though you're absolutely correct that the unprepared public, if you will, I mean, you can't expect all the public to be up on the scientific details here, but you can probably translate much of that into understandable terms that they can relate to and hopefully can then make up their mind. In the case, for example, of mRNA vaccines, um, you would think that in the final analysis, they would see the benefits, which in this case stare you in the face. It's not so clear with in vitro gametogenesis. This is a far more subtle and I would say elective decision. The mRNA vaccines are probably not even an elective issue. Nobody wants to contract the disease and take the risk of succumbing to it. In vitro gametogenesis is going to be a much harder sell, and I think a much more difficult proposition to tackle. But there is no other way, it seems. I think merely imposing those outcomes on the public is no longer acceptable, and I think for good reason. Hard as it may be to go through the process and make it all work.
Thank you. I see that we have Dr. Weinstein has a question. You can either say it, unmute, or type it. Okay, I don't see the question here yet. And I have another question. So do you think that the public should determine even what science should be done or how the scientific discovery is implemented? Because you could develop a scientific method to do something that the public does not want to use. Should we actually limit the path to even, let's say that the public decides that the in vitro gametogenesis is, should not ever be used. Should we still even limit the technology, which usually is impossible anyway, in such a large, global environment? But what do you think should be the path?
Again, I would think that the public could see the benefit of the clinical component here. Most members of the public obviously wish to have a family and wish to see others complete that dream if that became possible technically. Whether or not all the rest of it will appeal to the public is, of course, something we'll have to find out. But in general, I would say that the public, when brought in the right context and educated accordingly, will show the necessary compassion for their fellow men and women, simply because they understand what it means intuitively or sometimes in real time, what it means to be infertile or unable to conceive, or so I would think that what I call the Hippocratic beneficence component, the remedial opportunities that arise, probably will be acceptable to the public, but they may well place significant limits on other elements of the technology, particularly the ones that could potentially get out of hand. But your point about whether or not one can actually keep the genie in the box or not is a good one. And I'm not naive enough to think that this could be controlled and or maintained, if you will, under a tight lid throughout the globe. But all I'm saying is, let's point out the positive principles that we should deploy when and if this happens, and let's do the best we can. And we can probably barely account for what's happening in the United States, let alone everywhere else. So I'm not naive or incognizant of the limitations, but I'm still trying to put out the constructive and positive elements of the equation that I think an audience such as yours probably appreciates.
We have here several questions, important questions. First from Dr. Tia Powell, the Director of Bioethics at Montefiore, and she's asking, do you have any thoughts about how to regulate the enhancement overhang? Would we choose which genetic factors are possible to edit and which ones are not?
Again, excellent question, of course. And I think when I presented it, I already conceded to be at a loss as to what and how. But I would think that these issues, if I had anything to do with it, for example, in the context of a National Academy of Medicine report, I think we will try to probably come up with any and all scenarios, absurd as they may seem at the starting point, and try and logically and consistently make suggestions as to what is potentially permissible and what is not. None of these things, I don't have to tell anybody in this audience, is absolute. It's all very opinion dependent. And with the exception of some egregious extremes, I think reasonable disagreement between people can be anticipated. So no perfect answer, I'm afraid, but enhancement, if put to good use, I would say in general, could be blessed. On the other hand, enhancement that could be put to negative purposes, belligerent purposes, any purpose that is non-constructive, shall we say, clearly will be frowned upon or worse. So I know this is merely a statement of general guidelines, but that's probably as far as I can take it at this point.
We have a question. First, the person who is asking the question, who is anonymous, says, thank you for a wonderful talk. What about banking of in vitro gametogenesis and use in post-mortem gamete creation? What do you think about that? Science fiction, reality, another bioethical dilemma?
All of the above. I mean, the notion of us being perpetuated post-mortem is one I think we were all going to have to reflect on. But it's one of the complications here, just as I mentioned, the seeming impossibility of non-consensual parenthood. This could well be yet another example, consensual or otherwise, of parenthood, in this case, post-mortem, which I think raises issues that had never to be faced by the pharaohs or some of our other predecessors. So how do we approach this will require careful deliberation. I don't have an immediate opinion on post-mortem reproduction, if you will. But those are all interesting and provoking issues that will have to be addressed. It just points out yet again that when you open this Pandora box, all kinds of things emerge, and it would have to be a very thorough treatise when and if we get to deal with this. One thing I'm telling some of my friends at the National Academy is that for once, we may want to get ahead of the issue as opposed to play catch up. In other words, I don't want to read on the pages of Science or on the pages of the New York Times that in vitro gametogenesis has been accomplished in a human. I would rather deal with this beforehand if we can help it. And I think that message is gradually sinking. That has rarely happened, by the way. Gene editing was a post factum report. Mitochondrial replacement was a post factum report. There are many such examples. If we can do this before this actually happens, we would be far better off, and we would not be caught flat-footed, which we would otherwise be.
I have a question from Joshua Weinstein. How do the transforming factors interact with silenced genes and allow them to potentially be active? It's a technical question. I don't know if you want in 1 minute to address it.
I actually don't know the answer to that. I mean, it's a good question, but I'm happy to correspond perhaps to and dig deeper into some of the latest papers to see if we can figure that out. I'm certainly not up on the details at that level without further study.
And one last question from Dr. Scott Monrad. He's talking again about the enhancement overhang, as you call it, and is asking, have any religious bodies taken stand on the ethics of these technologies?
Good question. Nothing comes to mind, although it's rare that you, in the United States anyway, that you don't hear about these issues from religious quarters. This is, I think, perhaps at this point, not quite up there amongst the issues that most people are cognizant of. It doesn't exactly fit into the pro-life, pro-choice debate because it occurs at the level of gametes rather than embryos. But there are implications, obviously, to embryos and to life. And sooner or later, I suspect we will hear more. To my knowledge, that has not been a major area of preoccupation of the sectarian community in the United States or elsewhere. That's not to say that it won't be, but it stays clear right now, at least, of the issue of life, which technically begins with the embryo, although that's a debate for another day. But since we are limiting the conversation here to gametes, it's a very different discussion. And I don't think there has been that much that I've seen that focused on this issue from a sectarian point of view.
Thank you, Dr. Adashi, for a fascinating talk on a fascinating topic. I'm sure there are many more questions. And with that, we'll finish this grand rounds. Thank you again.
My pleasure. Thank you.